If I could describe a farmer in 3 words, hardworking would be number 1.
Farmers are the most creative people.
I think farmers are the most selfless people.
Reliable,
dedicated,
resilient,
passionate,
essential.
Hi everyone, and welcome to another Seed World Giant Views interview. My name is Alex Martin, and I serve as the editor of Seed World magazine. And today I am so happy to be joined by Beni Kaufman, who is Director of Operations at Agriplex Genomics. We are here at the American Seed Trade Association's very final year in Chicago before we move over to Orlando. But that is okay because Beni has been here all week with us. He gave a wonderful presentation yesterday at the Ag Tech Innovation Showcase, and today he's going to help us dive into a really fun topic, molecular marker technologies and how they are bettering our seed sector. Thanks so much for joining me today, Beni. Let's start with some introductions just for people who might not know you very well. Can you introduce yourself and kind of tell us more about your role at Agriplex?
Sure. So, I don't know exactly what's the best place to start, but I got my degree from University of Illinois. I think most of my career I spent with Pioneer, then DuPont Pioneer, and then who remember what's the rest of the names. Before starting with Agriplex Genomics, I did some consultancy, and before that I took a break from the lab and did some administrative work for Vista as the Secretary General. At Agriplex, I started as an operation director. I spent most of my time in developing SNE panels for specific crops or livestock, and then pushing those panels into the community. I'm not sure if pushing is the right word, and making it available for the industry and academia and public institutions.
That is perfect, Beni. What a great introduction. That is a vast history you have behind you. So I figured we could start broad today, and you could tell me a little bit more about molecular marker technologies. What is it? Why do we need to know about it?
Well, let's start with what is it. So people were looking into ways to probe the genome, get a glimpse at the source that is the DNA. And obviously at the time, I don't know, 40 years ago, if not longer, there was no way to do whole genome sequencing. The cost was prohibitive. The technology was too slow. So people came up with an alternative, which is basically ways to sample the genome. If I can't get it all, I can get a glimpse at certain, no matter how small, portions of it. And that's what molecular markers are. They are a way of sampling the genome.
That is a great introduction to what molecular markers are and why we need to be aware of them at the seed industry. I know molecular markers are not a new technology per se, but they are newer in history. How have they developed in recent history, and have we seen it get a little bit more advanced as customers start to use it?
Of course. So over the years, there were a number of such sampling methods, as I call it. I don't know. In the context of agriculture and breeding, I think it started with RFLPs, and then we moved to SSRs and Rapids and AFLPs, and endless versions of all of those. And then SNPs were developed or were recognized as a valuable way as a valuable molecular marker system. But what happened next was that the sequencing methods have developed. They become cheaper, faster, and then we had the possibility of doing a whole genome sequencing. But what is interesting is that once we did that, we realized, or people realized, that we don't need all the sequence. Much of it is not really useful. All the monomorphic parts of the genome, when you compare between lines or individual, they are not informative. So even now, when you can do sequencing in a very efficient and economical way, you're still using those SNPs, because they are the SNPs and indels, because they are the major source of DNA variation. So, if you will, it's a kind of a closing the circle. So SNPs most probably are the ultimate molecular method.
Excellent. And I'm glad we brought up SNPs, because I know that's becoming a more talked about technology within genomics. Now, can you tell me, why does molecular marker technology matter for breeding? What are kind of some of the benefits you're seeing using it in breeding programs?
Sure. So there, I think there was a period of time that was a topic in vogue in many articles. So what I'm going to say pretty much just touches the surface. But I think I can name a number of advantages. So first of all, obviously, the overarching title for using molecular markers is molecular breeding, which basically means that the genetic information, or as I called it earlier, the source, or the variation that we're able to interrogate, is used as the mean of making a selection decision or advancing decisions. And the advantage of doing this based on molecular markers comes from a number of things. First of all, you no longer need to wait for the plants to grow. You can make your selection decision at a very early stage. You can do your analysis or your genotyping at the first leaves, or even before that with embryonic leaves, the cotyledon, and that translates into efficiency and savings. You make the decision early, so you save on greenhouse space, you save on nursery space, you save on all the resources that go into growing those plants, labor, water, soil, fertilizers. So that is one obvious advantage. On top of it, you have the fact that you can have more intensive selection. So it takes part, not all obviously, it takes part of the guessing work out, and you have data that you can base your decision selection on. So the selection intensity is higher, and with that you can finish your program much faster in terms of number of cycles that you need to achieve your goals. And again, if you need to do 4 or 5 cycles using whichever method of marker-assisted selection, that is much shorter than 8 cycles that in traditional breeding or so, sometimes even more, it would take you to get to the same endpoint. And again, this savings in time translates to savings in resources, saving in money, obviously. It also shortens the time to market. And that can be interpolated further to more earning opportunities and, as I said, more saving. The last thing worth mentioning is that the molecular markers are giving you a good way to have a QA, QC throughout the whole workflow, starting from the point that the lines that are developed are making it into the hands of the breeder, throughout the breeding process, and further on when it's being handed on to the production or supply part of things. It is very important that along all this chain of events you're able to maintain or have a tap on the genetic purity of your materials. Molecular markers gives you a good way to do this and incorporate it smoothly into your other activities. You don't have to step aside and do a grow out and so forth. You can incorporate your QA, QC markers with the other markers that you're using, and you have a constant chain of custody, making sure that the genetic makeup of your breeding material or your production is not being compromised.
Perfect. And I want, within that answer, you kind of brought up the data that you're going to receive from these molecular markers. Is it ever overwhelming for seed companies to kind of deal with all of that data that comes out of using molecular markers for plant breeding? How do you manage it?
Sure, it is definitely a challenge. Just in parentheses, it's not only seed companies. Academians or people at the universities that are doing research on molecular breeding, which is equivalent to the one that's done in the industries, they also face the same challenge, and the same goes to public institutions, research institutions, or breeding institutions. So it is a big challenge, and the fact of the matter is that the challenge is actually starting way upstream, even before you generated your molecular marker data. It starts with the ability to identify and choose markers, and then taking this information in, and that is where for us at Agripex this challenge starts, and that is when you get the sequence information that a customer is interested in. You have to, and sometimes it's thousands of SNPs. You need to be able to take in the data and make an assay and develop primers out of it. Fortunately, we have a talented team of analysts and programmers. So they generate the software, and that is really the answer. So they generate the software that enables this. In our case, it's Plexform for primer design and assay development. And then on the back end, and that's what really you were talking about, when the data comes out of the sequencers, another set of programs under PlexCall that are taking the sequence information and transforming it into genotypes and or allele frequencies and so forth. And it goes the same for the breeders. It's all a programming challenge more than anything else. And that's why in parallel to molecular molecular markers development, you also have the development of bioinformatics, which is really the field that enables solving this data avalanche. Now, for seed companies, they have other options, and that is if you don't have the resources in-house to deal with that, there are you can outsource it, and there are companies like Energen or Delta G that will be happy to do the work for you.
And I just have one last question today for you, Beni. What are you excited to see in the future of molecular marker technology? I know there's a bright future ahead of molecular markers, but I'd love to hear your opinion on what you'd like to see in the new development stages.
Sure. So as far as marker system goes, I said earlier, I think the SNPs are the ultimate marker system, but the next step will be haplotype genotyping. So no longer looking at one singular discrete SNP to genotype, but if you will, look at the neighborhood. And that will be very informative even to the researcher that is trying to do association of traits to DNA segments in QTL studies or association studies. And that will be a great help for more challenging species or crops like all the polyploid crops. So the other part of evolution that I see is not so much in the development of how you generate the data, but the applications that the data is used for. So obviously the big thing in recent years is gene editing. I think that in an interview like this a few years back, I was talking about the need to develop systems to to test for gene editing, to confirm and to satisfy possibly regulatory requirements. And here we are. We already have the need to do edit confirmations. The demand of that is growing. So that is a new application of molecular markers, if you will. The regulatory is lagging behind, but it will come too, and that will require a lot of analysis. Another step in or another new application of molecular markers is in the new, well, relatively new field of soil microbiome. So this is something that the soil fauna and flora, microbial or unicellular and so forth, turns out to be a very important component in the ability of the plants to develop and yield. So people are now looking at what's in the soil, and that definitely can be done with the help of molecular markers. So this is something that we actually will be going to look into and offer it. And I think these are the two big things that come to mind at the moment.
Awesome. Well, I think that's a wonderful way to end our interview today, Beni. Thank you so much for joining me. And to everyone at home, thank you so much for listening. Of course, this is not our only Giant Views coming out from ASTA. We're on the floor all week, so make sure you stay tuned for any other Giant Views that are coming out from the show. And until then, we hope you have a wonderful day and that you've taken home something that you can talk to your family about at the dinner table. This is Alex Martin from Seed World, and we hope you have a great day.